Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. Now today I want to discuss a very interesting topic, that is biopolymer. But nowadays we have great concern, am I right, that plastics that polluted our atmosphere to a great extent, and biopolymer plays very important role, and obviously that coming in a they're slowly coming in a very big way. And as you know, the reason for that is that that polythene that is we use in the market, that is non-biodegradable. Mostly they are non-biodegradable. So when you throw it to the atmosphere, that remain as such. There is no degradation take place. So that is the problem, because that is the problem that now question comes how we can get rid of from that. And one of the way for get rid of this problem is the use of biopolymer. So let us discuss today how the biopolymer coming in the market. Now first let us try to find out that what do you mean by biopolymer, that what are the different biopolymers available. The biopolymers, they're the polymers produced by the living organisms. In other words, they are polymeric these biomolecules. I can give the typical example of this cellulose. Cellulose is a polymer of glucose. Am I right? Cellulose is a starch. As for example, starch is a polymer of glucose. Only the difference is that in the cellulose, the glucose bonding is alpha 1,4 and alpha 1,6 in case of starch. Where in cellulose we have beta 1,4 linkage. That is, but both are polymer of glucose. Then we have lignin, we have protein, all are polymeric compounds. Then known as the renewable polymer. Renewable polymer means they can produce again and again. Renew term means it can produce again and again. And I have given the cellulose is the most common organic compounds and biopolymer in the earth. And since they are polymer, biopolymer contains monomeric unit that are covalently bonded to form the large molecules. The polymer, I can give the example typical. Suppose we have the formula of glucose is C6H12O6. Am I right? So when we say polymer of glucose, then what will be the formula? C6H10O5 to the power n. Am I right? This is how it can be. This is how the polymer formula is looking. This is the n number of molecules. So one glucose molecule is binds with another glucose molecule like this. This is and this linkage is either alpha 1,4 or beta 1,4 or maybe it is alpha 1,6 linkage. Now there are three main classes of the biopolymer. If you look at, one is polynucleotides, that we have RNA and DNA, which is a long polymer composed of 13 or more nucleotide monomers. Then polypeptides, we have this protein molecules, we have the polymer of amino acids. Then polysaccharides, they are linear bonded, this is polymer of carbohydrate structure. So you know that if you see that three different types we have, one is polynucleotides, one is polypeptides and polysaccharides. And other examples of biopolymers is the rubber, suberin and melanin and the lignin. Now what is the classification of the this biopolymer? Biopolymer derived from biomass. This is biomass such as the agro residue. Examples are starch, lignocellulosic material, protein and lipid. I told you before also. And biopolymers obtained from the microbial production is the polyhydroxy alkaloids. alkanoids. So this PHAs in abbreviation form we call it PHA. And you know that this is we have been working with an organism called Enterobacter cloacae for a longer period of time for the production of hydrogen. This organism also produce this this kind of hydroxyalkaloids, because you know that this is the cell mass, and you know that this is the growth associated product. The more you have cell mass production, this is polymer production is there. So this is like this, and biopolymer conventionally and chemically synthesized, the monomers are obtained from the agro residue like the polylactic acid, or we call it PLS. This is lactic acid, all of a sudden that we find it the tremendous industrial potentiality. And why it has industrial potentiality? Due to the development of polylactic acid. And what is the use of polylactic acid? Because it can be used as a in case of in the medicinal sector to a great extent. You can imagine that in case of internal surgery, in the early cases, that most of the operations doctor, the surgeon usually use the nylon thread for the stitching purpose. Now they use the the kind of clip made of polylactic acid. So you know that can absorb in the system automatically in a long time operation. This is a tremendous potentiality we have in the market. Biopolymers whose monomers and polymers are obtained conventionally by chemical synthesis, such as the aliphatic and aromatic hydrocarbons. So these are the different classification that we have as per the biopolymers are concerned. Now let us look at the property of the biopolymer. First, this is the renewable. Renewable is very important because this will be available with us throughout our existence. That is renewable. That means it will not be exhausted. Now, sustainable, it can be with us for longer period of time. Biodegradable, which is very important, because if it is biodegradable, then it is environmentally friendly. Then it is non-toxic, it not causes any kind of hazard, and non-immunogenic that we have, then non-carcinogenic, that doesn't cause any kind of cancer, and non-thrombogenic, that it doesn't causes any kind of blocking of the blood flow, and carbon neutral. So you know that there are tremendous that if you look at the properties that indicate that is the ideal that things that we have for our use in the day-to-day life. Now, if you look at the application of the biopolymers, there are several that we have. It can be used as a coating, because sometimes as an apple, sometimes we coat with kind of polymer, so that the freshness of the fruits will remain. Then fibers and plastics and rubber, it can be used as an adhesive, it can be used as a cosmetic, it can be used in oil industry, paper, textile industry, water treatment, biomedical, and the pharmaceuticals. So if you look at different applications, it has tremendous potential. It doesn't have one or two applications, it's several applications. So we can use this biopolymer in the day-to-day life. So question again raised that why we are interested for the biopolymer. I told you that one important reason that this biopolymer is that it is renewable, that it can be sustainable, it can be with us for longer period of time, carbon neutral, and the renewable, and mostly they are made from the plant material, which can be grown indefinitely. Biopolymers are biodegradable, and some are also compostable, and they are broken down into the carbon dioxide and water by the microorganisms. Biopolymers has low environmental footprint. The petrochemical based polymer pose severe environmental pollution problem. Replacement of these chemicals by biopolymer will safeguard our environment to a great extent. So this is the benefit that we have with the biopolymer. So there is no doubt that why we should not go for biopolymers. Then we should go all the way to the biopolymer. Only the thing is that we still have to look into it should be cheap. It should be so that we can use easily for our day-to-day life. The uses of biopolymers, I told you that we have polylactic acid, natural occurring gene, and polyhydroxybutyrate, and this can be used as a plastic replacing the need of polystyrene or polyethylene based plastic. Gene is a class of this polyprolamine protein found in maize, and it is usually manufactured as a powder for corn gluten mill. This is the kind of application that we have with the gene. Let us talk something about the polyhydroxy alkanoids, because this is largely we have produced in the market, and this polyhydroxy alkanoids, this is PHAs, all polyhydroxy alkanoic acid, the main kinds of biodegradable and biocompatible polymer. Main we target because we find lot of microorganisms, they can produce this particular polymer. BHAs can be produced by different bacterial cells, and BHAs are the first biomaterial discovered by the French microbiologist M. Limogens, that using the bacterial strain Bacillus megaterium. This is the Bacillus megaterium they found. This can be used for the formation of this PHAs. And they are classified as a linear polymer polyester, and typically the PHAs can be produced in nature by several microorganisms such as yeast, fungi, and most of the bacterial strain. I just pointed out that we are using the Interbacter cloacae for the production of hydrogen for last couple of years, and we find this organism also produce this this polymer, this kind of PHAs to a great extent. And if you look at the chemical formula of this molecules is given here. Now question comes how the production take place, how the PHA production take place. This is from the simple sugar. This is the TCA cycle. It comes to acetyl CoA, then acetoacetyl CoA, then hydroxybutyl CoA, then this produces PHA. Other way, this also there is another way you can produce from the carbon source the fatty acid, again acyl CoA. This also derivated to this. Another is the carbon source sugar, acetyl CoA, manual CoA. This also can produce PHA. This is how in several way we can produce the PHA. The industrial production of this PHAs can be taken place by the using of different microorganisms as listed here. You can see that A. eutrophus, that different type of organisms, Pseudomonas species we have, Halomonas that we have, FedEx species we have, Aromonas species we have, then there are different Thermophilus, there are different type of organism, E. coli, or recombinant E. coli also there, Chromobacterium species, activated sludge also here, cyanobacteria. So different type of organism can be used for the production of PHAs. Now question comes that what are the substrate used for this PHAs production? We can use the different type of substrate for the PHAs production. Sugar the sugar beet molasses, sugar cane molasses, sugar cane liquor, the soy molasses, hydrolyzed soy, the corn strip liquor, glycerol, fish protein media, casein hydrolyzed, urea, cheese whey, hydrolyze starch, cellulose hydrolyze it, sucrose, xylose, galactose, mannose, arabinose, glucose, fructose, rice bran and cornstarch, sugarcane bagasse hydrolyze it, lactic acid, oleic acid, acetic acid, sunflower meal hydrolyze it and olive oil, etc. So, different material can be considered as the substrate for the production of PHAs. Now, if you look at the flow diagram, that how it produces. So, this is here we know that how we industrially produce. We have a strain. Suppose we have this microbial strain in a slant. Then we put it from this. We put it in a conical flux. This is the conical flux. We grow liquid media with it. Then we transfer to the reactor. Then slowly increase the volume, and we come up with the production fermenter. And after that, we separate out the cell through the filtration process. Then we do the drying. Then after drying, we do the powdering. Then we do the extraction of that polymeric material as extracted out. Then we do the ultrafiltration, and then after that again we PHA precipitation is there. Then centrifugation. Then again we dry, air dry, and then we pack it in a container and sell it in the market. So this is how this PHA's production take place in the industry. That we started, it is similar to the, I told you that it is a growth associated product as the cell mass production. So we encourage the increase in the cell mass production, so that that our PHA's production can be increased to a great extent. Now these are the different how the effect of nitrogen source that has been shown here, that the different type of carbon source, glucose, sucrose, sorbitol, mannose and sodium glutamate. But now this nitrogen source we have yeast extract. Now you see that if yeast extract is used in case of glucose, the pH's content is about 45.2%, but urea in case of it is drastically reduced 14.8%. In case of ammonium sulfate it is 29.3%. Now when we use sucrose, we have found that different effect that we have, and then in case of sorbitol we find that this extract gives a higher amount of this BHA production, and then urea also gives the significant amount of PHA production. Now in case of mannitol we find the maximum the production of PHA take place by using yeast extract. Then we have ammonium sulfate. Then we have this urea. The sodium gluconate, in case of that also significant amount of BHA production take place by using yeast extract, but here maximum production take place in case of using ammonium sulfate. Regarding the factors affecting the fermentation process, the team and Sten Buchels that reveal that the production of PHA by Pseudomonas aeruginosa, that it was found that when the cell reaches the stationary phase, the intercellular PHA was decreased. So that we know that we know the cell growth cycle is like this. So maximum production take place during this log phase. This is the log phase where the maximum production take place. Now Logiven et al. In 1998 produces PHA from normal octane by Pseudomonas oleoborans, and it was found that the microorganism has a specific duration of BHA recovery. However, only some strength accumulate the PHA along with the growth and growth associated product, because most of the when cell grow, we find that in the cell mass comprises of this PHA itself. The mineral substance, how it affects the fermentation process. Mineral substrate like phosphorus, sulfur, magnesium, etc. could affect the microbial growths in several functions. And phosphorus is assimilated only in the form of orthophosphate, and it is essential for all organisms. Sulfur is incorporated as a S containing sulfuric amino acid in protein, and magnesium function as cofactor and effector of many enzymes in the microbiome. This is the general things that is applicable to the microbial system. And pH and temperature, this is also as usual, that plays the very important role in this fermentation process. Normally the pH usually carried out under normal moderate temperature 30 to 35 degrees centigrade, and pH is 6.5 to 7. This is the condition that we have. Now the polyhydroxy alkanoids that has the different applications. You can see here that in plants and scaffold that is can be produced, drug delivery system it can be used, agricultural is that used, packing material is can use, tissue engineering it is used, it can be used as an energy source. The various application we have as per PHA is concerned. Now the biodegradability of PHA is a very important factor, because in the plant we have the carbohydrate, and this carbohydrate when we use in the fermentation process is produces PHA polymer, and this is plastic product, and this is biodegradable. You can recycle or biodegrade it to carbon dioxide and water, and this again through the photosynthesis process in the plant we produce. This is a cycling device that we have in the system. Let me discuss something about the polylactic acid. I told you that lactic acid nowadays has tremendous potentiality in the market, and PLA is a thermoplastic aliphatic polyester. Polyester that that the PLA is the thermoplastic aliphatic polyester biodegradable polymer that derived from the renewable sources by means of fermentation process using sugar from corn, followed by either ring opening polymerization or by condensation of polymerization of lactic acid. So this lactic acid undergo polymerization reaction, and then we can produce this lactic acid. This is the formula of lactic acid, and lactic acid formula is like this: CH3 CHOH COOH. That this lactic acid they can produce this polylactic acid, and it looks like this. It is perfectly clear. You are going to see this is perfectly polymer. When you we extract it, is perfectly clear. Now, general property of this polylactic acid is, first is the transparency. We have already seen that it is quite transparent. The glass transition temperature is 50 to 60 degrees centigrade. Melting point 130 to 180 degrees centigrade is quite high. Crystallinity is 30 to 40 degree, 40%. Tensile strength is 4 to 6 kg per square millimeter square. Elongation is 3 to 4%, and flexural strength is 7 to 9 to 11 kg per square meter. Now PLA is considered both biodegradable and biocompatible in contact with the living tissues, and for biomedical applications such as implant and drug encapsulation, etc. Just I pointed out that medical sector is largely used this PLA polylactic acid is largely used in the medical sector, and PLA can be degraded by the abiotic degradation, simple hydrolysis, the ester bonds without requiring the presence of enzyme to catalyze it, and environmentally friendly material. So this is the advantage of using this polylactic acid. Now production of polylactic acid is like this, that this we can produce from the corn. and sugar beet. You can see that this has the raw materials. Then we take in the fermentation process, and then we have lactic acid. Now here I want to point it out that lactic acid can be produced also from the whey. Whey is a byproduct of the cheese making industry, and we, as you know, that cheese is a kind of product through which we can preserve the milk, protein and fat for longer period of time. So we have lot of cheese consumption throughout the world. So we have a lot of whey production, and the whey is the best raw materials for the lactic acid production. Besides the whey, that we considered that corn, and if you look at the corn and the sugar beet also can be used in the fermentation process for the production of lactic acid. And this lactic acid undergo condensation, then polymerization, then reforming, and then forming, then we have the product. So there are several steps involved before we get the polylactic acid production process. So this is how we can form this polylactic acid. Now this is the application of polylactic acid. You can see that it is rigid, that if it is rigid it can be used as a container. This is a different type of containers you can use, and you can use as a food serviceware, that different food product, giving the food products we can use here also. The non-oven fibers that we can use here, the non-oven fibers we use, and durables, different the materials we use in the lipsticks and other things also we can use. So that I want to point out that it has different uses. It's not only one uses, it's the different uses that we have here in case of polylactic acid. So in this particular presentation I try to point out about the potentiality of biopolymer in our market, in the day-to-day market, because we find that the plastic which poses a lot of environmental pollution problem, because the reason is that the plastic is non-biodegradable in nature, and it causes a lot of the hazard. As for example, particularly we have seen in the water line, when we throw this plastic in the water line, it choke our lines and doesn't degrade. So it is a severe problem. And one way we can solve this problem, that by using biopolymer, and biopolymer we have, that is largely the marketed in two different form. One is PHA, another is PLA. PHA usually produced through the microbial fermentation process, and PLA also produced first you produce lactic acid, then we through the chemical process we make the polylactic acid. So that this is how we can produce, and advantage of this particular biopolymer is that they are renewable. They are produced from the material which is largely available with us, and that will not be exhausted, that will be produced again and again. So we can keep on producing those material, and it can be biodegradable. Since this is biodegradable, it will not causes any kind of environmental hazard, and since it is not posing any kind of environmental hazard, so it will safeguard our environment to a great extent, which is very much desirable. And particularly I told you that that polylactic acid has tremendous potentiality in the medicinal sector, particularly pharmaceutical purpose, that particularly operations and other things, doctor largely they use the polylactic acid materials, and also in some food products and other things it is used. Only the constraint of using this is the cost of production. If we reduce the cost of production, I am sure that in near future we can replace our plastic by this biopolymer to a great extent. Thank you very much.